Welcome everyone, my name is Zsolt Holló and I would like to present you the PREDYSTIC Infliximab RA complementary diagnostic test. So rheumatoid arthritis is a global problem affecting almost 1% of the population, and the therapeutic approaches are based on mostly trial and error selection of drugs using first, second, and third line treatments of synthetic and biological disease-modifying agents. And we wanted to apply the concept of personalized medicine in the practice for rheumatoid arthritis patients and the drug selection for these patients, based on the fact that we have seen differences or heterogeneity of the rheumatoid arthritis patient populations, and we wanted to select non-responder and responder patients concerning the drug response. So almost 1/3 of the patients do not respond to biological therapy, and currently there are no biomarkers or diagnostic tests available to select the first line biological therapy. So we started to work on this problem and decided that we shall develop a peripheral blood-based response prediction test, which can predict the disease activity and the specific disease activity score, the so-called DAS28 CRP value. So in this way, we can have the clinician, and the clinician can stratify the patients as responder or non-responder patients, and in our case the responder patients can get infliximab and the non-responders can go for an alternative biological therapy. So our diagnostic test performance surpasses the current trial and error based approach. So if you take a look at the accuracy values for this 1/3 responder patient, we can calculate with the 35% accuracy, and this can be significantly increased to 88% if we apply our testing approach. And as you see, the other conventional diagnostic parameters like specificity and sensitivity reach meaningful values of 94% and 79%. The test development followed the typical pathway of developing diagnostic tests. So first we started to work on our biomarker concept based on gene expression profiling. We have identified a large number of biomarkers, so 250 biomarkers were roughly identified. And we have also selected a robust test method. We have also performed a diagnostic platform change, and afterwards we could complete our analytical performance validation and the clinical performance validation. And once we have all completed these phases, then we could move on to CE IVD registration. So this is a clinically validated in vitro diagnostic test. We have set up a multicentric clinical trial in 7 countries at 32 sites, and we have enrolled 217 bio-naive rheumatoid arthritis patients with moderate to high disease activity. This was a non-interventional study, so this means that we were not interfering with the routine care for these patients. And as you see here, our patients were followed for a 1-year period with 4 visits from month 0 to month 12. And during these visits, the clinician is typically evaluating the effectiveness of the therapy after 3, 6, and 12 months, and they can make a decision whether to continue with an effective therapy or switch to another biological drug in case of non-effective therapy. So we perform our testing at 0 time point, and based on that, we would like to predict the disease activity score and the infliximab decision at month 6. So this is our concept. So this is a typical representation what could happen to a patient. So this is a disease trajectory and how we predict the disease activity. As you see here, we show here the so-called DAS28 CRP value as an index for the disease activity, and 3.2 is a cutoff value above which we can consider the patient as moderate, having moderate or high disease activity, and below this value it's a low disease activity range. And the patients can follow, after initiating the infliximab therapy, they can follow various routes. So in ideal case, they go below this 3.2 value and achieving a responder status. Non-ideally, the disease activity remains relatively high, and in this case, we can stratify the patient as non-responder. And so by using PREDYSTIC, we can predict the specific value and also this categorization of non-responder and responder. So how we moved on from the initial concept to the biomarker discovery and the modeling. We wanted to look for various patterns in these patients based on gene expression profiling. And we have selected an initial set of biomarkers and clinical parameters. And of course, we were looking for those markers which predict well the biological drug response. We have also applied machine learning and artificial intelligence during the analysis of the learning and training cohorts. And we had to analyze a huge data set in terms of various models. So therefore, we turned to this technology. And we moved on, of course, testing the various model candidates on the test cohorts, and cross-validated these early models, and a limited number of models were selected for clinical validation on an independent patient cohort at the end. So we use Real Time PCR platform and gene expression profiling combined with our proprietary algorithm, and we ended up with a relatively high complexity diagnostic test. So we utilize almost 50 genes, additional clinical laboratory data, and some clinical parameters. The platform is the Tacman low density array card platform, which is a microfluidic card hosting samples for 7 patients. And the instrumentation behind is the QuantStudio DX from Thermo Fisher. We not only developed the test itself, but we have also worked on the test workflow and the central laboratory service model. So at the sampling site, we can use PEX gene or RNA guide tubes for taking the blood samples. These samples can be shipped to the central laboratory for testing, and afterwards the data can be analyzed and validated using a cloud-based solution, and the physician receives a test report with the specific parameters for the patient. So this is a certificate of our test we have received from the Hungarian authority. So we are CE certified for whole Europe. So in summary, the PREDYSTIC Infliximab RA test is a precision medicine tool which can optimize the biological therapy selection for rheumatoid arthritis patients. And this is a clinical decision support for initiating the biological therapy, and we can predict the therapeutic response at month 6 to the specific biological therapy. The test is based on blood sampling, and we have received the CE mark for the test back in December 2020. So how we are making impact? Of course, we have started to present our data at various conferences. We have set up a LinkedIn showcase site for the presenting our new information and new data. There's also a PREDYSTIC microsite within Egis Health site. And with our partners, we have worked on various press releases, and we also presented our data and the concept at the Microsoft Envision Forum recently. So this just indicates that we are continuously sharing our new data and regulatory and other developments regarding the test. Right now, we are in this production phase. So we have performed various manufacturing steps until we reach the production for the market phase. And we are manufacturing our samples for potential license-out partners, looking for license-out collaborations and other steps to roll out the service to our patients. These are a couple of takeaway messages we have received from our partners during our discussions with potential license-out candidates. So as you see here, it seems that based on their evaluation, we have completed our development task, and we could cover really the whole value chain and achieve the good result in terms of product development and registration as well. Thank you for your attention.